Hey everyone, this is Devishis. Welcome to Biosynthesis. In this session, we're going to discuss some of the questions from TIFR 2021, and it is the second part of this series. So here I'm with the first question: which of the following statements are true? The first one is Vmax of an enzyme remains constant with increasing enzyme concentration, which is a wrong statement because we know that Bmax is directly proportional to total enzyme concentration. So that means if at all the Vmax will increase, sorry, if at all the enzyme concentration will increase, the Vmax will automatically increase because it's directly proportional to it. So it will not remain constant, it will increase, right? So Km of an enzyme for a substrate remains constant with increasing enzyme concentration. So Km will be remain constant. This is a true statement. Km basically the affinity between enzyme and the substrate, right? So it will remain constant throughout this process, whether we are increasing the enzyme concentration or decreasing the enzyme concentration. It is going to be constant. Km of an enzyme for a substrate increases with the increasing enzyme concentration. No, because this is a contradictory statement of 2. Both Km and Vmax of an enzyme remains constant. No, the Vmax will be increasing. I have already discussed in the statement 1. So the statement 2 is the correct answer, right? One DNA strand is attached to another DNA strand by. It's a very simple question. This is by the hydrogen bonds between the bases. Okay, so remember this, the bases. So bases are going to make the hydrogen bond. The adenine with thymine, the guanine with cytosine. There's a 2 bonds, 2 hydrogen bonds, and here is a 3 hydrogen bonds. What are the deamination products of cytosine and 5-methyl cytosine respectively? So if at all we'll deaminate the cytosine, so here is the cytosine, so we will get, see, uracil, right? The amine is here, the amine group is not here. Got it? So uracil and the 5-methyl cytosine, if at all we'll add a methyl group in the 5th position, what will we get? See, thymine, right? So uracil and thymine. Making a protein from a gene in eukaryotic cell involves, basically the question is given you for the translation process. From the gene, like from DNA, it will form the mRNA and protein. So this complete central dogma, the question covers complete central dogma. First, the transport of mRNA, then its processing, then transcription, then translation. No, the transport will not occur first, right? First, transcription of RNA is correct. It's transport, then processing, then translation. No, the processing will occur first. First, translation of the RNA, then its transport, then its processing, then transcription. Translation will occur in the last position. So we'll get the 4th one only. First, transcription of RNA, correct. Then its processing, yes, the splicing, the modifications, 5' modification, 3' modifications, then its transport, yes, then translation. So this is the correct sequence. The 2020 Nobel Prize in Chemistry was awarded to Jennifer Doudna and Emmanuel Charpentier for their work on developing CRISPR-Cas system for gene editing. This system is naturally. So basically it is an antiviral defense mechanism found in bacteria. You know, the bacteria is infected by the bacteriophages. So phage DNA sometimes in the lysogenic cycle integrated into the host DNA, in the bacterial DNA, in the lysogenic cycle. So that phage DNA will act as a guide RNA and it will give the antiviral immunity to the bacteria. So that is the discovery of this CRISPR-Cas9 system. Next, at which mitotic stage can standard karyotyping be formed using photomicroscopy of cells? So see, guys, in metaphase, you can see the metaphasic chromosomes. And if at all you are doing the banding, we can see the metaphasic chromosome is a very elegant way, right? So you see, it will be very prominent. The structure is very prominent and that is used for the karyotyping. So it will be metaphage because it's a photo microscope. You have to take a picture. So karyotyping is used the metaphasic stage because you can see very prominently these particular chromosomes in that stage. It's a very basic question. Which of the following amino acids do you expect to find in higher proportion within the histones compared to cytoskeletal protein? Very simple question. So guys, you know, histones helps in the packaging of DNA. So DNA is very long. Histones are going to bind with the DNA and it is going to pack the DNA. It is going to form the chromosomes, so like we have seen in the metaphase in the last question, right? So DNA are negatively charged. So we need a positively charged amino acid because histone is a protein, right? So here lysine and arginine, they are going to help in the formation of the histones. A human cell passes through 4 phases called A, B, C, and D. Each phase lasting for different lengths of time. At the end of the D cells, cell returns to A to continue the cycle. So let me write it down. So A, B, C, and D. Then again A. Then again the process continues A, B, C, D, right? In an experiment, a static snapshot of 10 to the power 6 unsynchronized cells. So what is the meaning of these unsynchronized cells? In a test tube or anything, a Petri plate, you have unsynchronized cells. That means you have mixed of different phases of cells. Like the A will be there, B will be there, C will be there, D will be there. There will be no differentiation. Everything will be present over there, right? And the total number of cells, total number of cells is equal to 10 to the power 6. So we can write it 2, 3, 4, 5, 6. It's a very easy way. And it was found that 3 lakh cells were in phase A. So in phase A, there will be 3 lakh cells. So if it all will count, it is 30%. It will come around the 30%, right? In a phase B, there will be 4 lakh cells. So phase B, it has 4 lakh cells. So it will be 40%, very simple. In phase C, it will be 2 lakh. So it will be 20%. And rest in the phase D, it will be 1 lakhs. Right, so it will be 10%. Okay, we got this. So then, the total time spent by one cell in the 4 phases is known to be 24 hours. So the A plus B plus C plus D is equal to 24 hours. Total 24 hours. Okay, so the phase A will contain the 30% of 24 hours, obviously. So it will be 8 hours. Phase B will be 9.6 hours. Right. So the phase C, how much hours it will contain? Is a 20% of 24 hours. Right. Phase C is a 10%, so 2.4 hours. It's 4.8 hours. Got it? So if at all you calculate the whole, then we'll get the 24 hours, isn't it? So what are the statements is given? The time spent in phase A is 5.2 hours. No, it's wrong statement. The time spent in phase B is 9.6 hours. This is correct. The time spent in phase C is 3. No, it's 4.8 and this is 8 hours and this is 4.8 hours. The time spent in any one phase cannot be obtained from given information. No, we have obtained it, man. Okay. So I hope this calculation is understandable by you. I hope you understood this calculation. Thank you so much for being a great audience. Please don't forget to like this session, share with your friends. Please hit the bell icon so that you could get updated about all the upcoming sessions. And please share with your friends and please do support me if at all you are liking these sessions, liking these videos. See you guys. Toodles. Bye-bye.